All right, welcome to the Explainer. Today, we are going to decode the master script, the very code that defines every single living thing on our planet. So, let's jump right in. How does life write its own story? Well, we're going to explore these truly remarkable molecules that carry the blueprint for everything alive, nucleic acids. So, you're looking at the very architecture of life. These instructions, they're not just floating around. They're neatly stored in these super long chain-like molecules called nucleic acids. And their main job, it's all about carrying information and passing it down, from one generation to the next. It's the ultimate hand-me-down, ensuring life continues. Okay, to really get a handle on this incredible code, we've got to start with the basics. We have to learn its alphabet. And that alphabet is built from one fundamental unit, a little piece called a nucleotide. So what is a nucleotide? The easiest way to think about it is like a single letter in this enormous genetic alphabet. And it's the specific order of these letters, the sequence, that spells out all the instructions for building and running an organism. Here you can see how it all fits together. It's made of just three simple parts. You've got the phosphate group, a 5-carbon sugar right in the middle, and then the all-important nitrogenous base. Now, what's really fascinating is how tiny little changes in these parts create the two different kinds of nucleic acids we're going to talk about. Okay, so the huge difference between DNA and RNA, it actually starts right here, with this one tiny detail in their sugar. If you look really close at that 2' carbon, see it? RNA sugar, which is called ribose, has an oxygen atom hanging off of it. But DNA's sugar, deoxyribose, deoxy literally means without oxygen, is missing it. That one little atom makes a world of difference for how stable these molecules are and what they can do. Now, these nitrogenous bases, these are the real stars of the show. They're the part that actually carries the information. They come in two families, if you will. You've got the purines, adenine and guanine, which are these bigger double-ring structures. And then you have the pyrimidines, which are smaller single-ring structures. Here's a little trick to remember them. The longer word, pyrimidine, actually corresponds to the smaller structure. Kind of counterintuitive, right? So in the language of DNA, the entire genetic alphabet is made of just 4 letters, A, G, C, and T. That's it. Now, RNA is a little different. It swaps out that T, thymine, for a slightly different base called uracil, or U. And the sequence of just these 4 letters is what spells out, well, everything. All right, so we've got our letters. Now, how do they get strung together to create probably the most famous molecule in all of biology? I'm talking, of course, about the DNA double helix. Okay, so the individual nucleotides are linked up one after the other by these really strong bonds called phosphodiester bonds. This creates what we call a sugar-phosphate backbone, and it's directional. It runs from what we call the 5' end to the 3' end. This direction is absolutely critical because all the machinery in the cell that reads this code is designed to move along the strand in this one specific direction, just like you'd read a sentence from left to right. And that brings us to the legendary Watson-Crick model. You see, DNA isn't just one single chain. It's actually 2 chains that run in opposite directions. We say they're anti-parallel, and they coil around each other. The strong sugar-phosphate backbones face the outside, kind of like a protective railing, while the precious bases, the information, are tucked safely on the inside. And the structure of this helix, wow, it's incredibly precise. It's so uniform. For instance, the distance for one complete 360-degree turn is exactly 34 angstroms, and an angstrom is a ridiculously tiny unit of measurement. We're talking about 1/10 billionth of a meter. And packed within that single twist of the helix, you will always find exactly 10 base pairs. They're stacked neatly on top of each other, forming the steps of this incredible spiral staircase. And finally, the diameter of the helix is also perfectly consistent. It's a uniform 20 angstroms wide. This amazing regularity is what allows DNA to store information so reliably for so long, and it's all held together by one simple, beautiful rule. And here it is, the secret sauce. The 2 strands are held together by a very strict pairing rule. Adenine, or A, always pairs up with thymine, T, and they're connected by 2 hydrogen bonds. Meanwhile, guanine, G, always pairs up with cytosine, C, forming a slightly stronger connection with 3 hydrogen bonds. This specific pairing is the absolute key to DNA's stability and, as we'll see, its ability to be copied perfectly. Okay, so if DNA is the master blueprint for life, how in the world does an organism make a perfect copy for every single new cell it creates? Think about it. Trillions of cells in your body, each with the same blueprint. Well, the secret, amazingly, is built right into the structure itself. Because A always pairs with T and G always pairs with C, the two strands are complementary to each other. That means if you know the sequence of one strand, you automatically, without even thinking, know the sequence of the other. And this is just a brilliant illustration of how it works. The cell literally unwinds the parent DNA molecule, and each of those original strands serves as a perfect template to build a new complementary partner. The result? You get two identical DNA molecules. But here's the cool part. Each one is a hybrid, containing one of the old original strands and one brand new one. We call this process semi-conservative replication. Semi for half, because half of it is old and half is new. Okay, so storing the code is one thing, but that's kind of a passive job, right? It's just sitting there. How does the cell actually read the instructions in its DNA to go and build the proteins that do all the actual work in the cell? To do that, to actually read the code, the cell uses a related molecule called RNA. And as we've hinted at, it's different in three really important ways. First, it uses that ribose sugar. Second, it uses the base uracil instead of thymine. And third, it's usually just a single strand. Now, these differences make RNA a lot less stable than DNA, which is actually perfect for its job as a temporary disposable message. It's like a sticky note instead of a book carved in stone. This all leads us to what is known as the central dogma of molecular biology. It's a foundational concept, and it describes how genetic information flows inside a cell. You can think of it like a one-way street. Information goes from the permanent storage in DNA to a temporary RNA copy, which is then used to build a protein. That very first step, going from DNA to RNA, has a name: transcription. So during transcription, this amazing enzyme called RNA polymerase latches onto a gene. It unwinds a little section of the DNA, and then it gets to work, building a complementary copy using RNA building blocks. Basically, the cell is making a temporary photocopy of just the specific instructions it needs right at that moment. But this RNA photocopy, which we call messenger RNA or mRNA, it doesn't work alone. It's part of a team. It works with two other types of RNA. You've got transfer RNA, or tRNA, which acts like a tiny delivery truck, bringing in the right amino acid building blocks. And then you have ribosomal RNA, or rRNA, which is a key structural part of the ribosome itself. That's the cellular machine that actually builds the protein. And there you have it. It's just incredible, isn't it? From just 4 chemical letters, you have a simple code stored in this beautiful, stable helix that can be copied with unbelievable accuracy and then transcribed into these actionable messages. This elegant molecular system is the universal language that connects every single living thing on Earth. So the question that's driving science now is, since we've learned how to read this code, what other secrets is it still waiting to tell us?